Hi friends, biomolecules are very important constituents of our cell, and we know that they are basically polymers which are formed by combination of monomers. Guys, protein, which is a very essential biomolecule, is formed of monomers known as amino acids. And we have also discussed that amino acids join together by peptide bonds to form proteins. But guys, how exactly the amino acids join together? And how are the peptides formed? And what are the different structures of proteins that are formed due to joining of the amino acids? Well, this is what we have to find out in this interesting session. So guys, the amino acids condense together to form peptides. And peptides are nothing but short chain of amino acids. So as I said, they condense together. Thus the reaction is a condensation reaction in which water molecule is removed during the formation of peptide. And how do they join? Yes, of course, I think you know it. It is by the peptide linkage. And the peptide bonds are actually formed when two amino acids come close to each other and condense together. How do they join? Well, we know that in amino acids, we have amino group which is responsible for the name amino acid. And we have a carboxylic group which is responsible for the acidic nature. So suppose I have two amino acids here. So amino group of one amino acid will join with the carboxylic group of another amino acid. And resultantly, a water molecule will be removed out and we'll get a peptide bond between the two amino acids. The peptide bond is the NHCO bond. So, as a result of peptide linkage, the peptides are formed. Now friends, if only two amino acids join together, then the resultant peptide is called dipeptide. If suppose three amino acids join together, then the peptide is a tripeptide. But if around 20 to 25 amino acids join together, then what we call is oligopeptide. And then comes the polypeptide. Guys, polypeptide has many or numerous amino acids which are joined together. So, the smallest peptide is the dipeptide, while you keep adding amino acids as many as possible into the dipeptide and you will get a polypeptide, which is the linear polymer of amino acids. Now friends, here you must be thinking, is the polypeptide same as protein? Well, they are same and the terms can actually be used interchangeably, but the condition here is that a polypeptide should have around 50 amino acids to qualify as a protein. Now the peptides that are formed, are they neutral or do they have some nature associated with them? Well guys, you have an answer in the brain, but you have to think over it. So, we know the peptides are made of amino acids, right? Now amino acids, if you remember, had side chains which were responsible for the nature of amino acids. So similarly, the side chains of amino acids can be acidic, basic, they can also be neutral. And consequently, the nature of peptide is actually decided by the amino acids which are present in the peptide. Now, after the peptides, what comes next? Yes, exactly, the proteins. So friends, what are proteins? They are heteropolymeric biomacromolecules which are made of one or more polypeptide chains. So different polypeptide chains, they combine together to form what we call protein. Now, what is the role of protein in our body? Do I even need to tell you that? Yes guys, they are the main building blocks of any living system. And also they are the most abundant biomolecules in living systems. And there are variety of proteins. So proteins are diverse in their structure and also in their function. There are variety of proteins exhibiting different structure and consequently different function. So in a cell, there can be thousands of proteins together in a cell performing different functions because of their different structures. So friends, the proteins are made of one or more polypeptides. Now those proteins which have just one polypeptide in them are known as monomeric proteins. Example is the muscular protein myoglobin. Those proteins which have two polypeptides, they are called dimeric proteins, like the insulin. Now those proteins which have four polypeptide chains in them are the tetrameric proteins, like the hemoglobin of blood. And the proteins which have numerous polypeptides or many polypeptides in them are the multimeric proteins, like the rubisco, pyruvate dehydrogenase, etc. Now friends, all these proteins are composed of amino acids which are linked together by the peptide bond. Now can you tell me how many amino acids do we have? Yes, exactly. There are just 20 amino acids. Then how do these 20 amino acids form so many proteins? That is why I said that proteins are diverse. So friends, these 20 amino acids, they combine together in different combinations to form proteins. And this is similar to the alphabets. So we have limited number of alphabets and we can form variety of words using just these alphabets. Similarly, using just the 20 amino acids, we can make a number of proteins. Also friends, the sequence of amino acids is very specific for a particular protein. Guys, collagen is the most abundant protein in the animal world. And rubisco, which is ribulose biphosphate carboxylase or oxygenase, it is the most abundant protein in the plant world and also in whole biosphere. Now friends, as I said, the proteins have specific structures and they show diversity. So guys, basically proteins in their structure have 4 levels of organization. So proteins show 4 types of structural organizations, which are primary structure, which is nothing but a chain of amino acids. Then secondary structure, which is formed by folding of the polypeptide chain to take either the shape of helix or the sheet. Then there is a tertiary structure, which is a 3-degree structure, and it is actually the 3-dimensional spatial arrangement of the polypeptide chain. And then there is a quaternary structure, which is made of multiple polypeptide chains joined together by various interactions. Now friends, all these 4 structures are formed because of the bondings which are present in the molecules. So there are various interactions which lead to formation of these bonds, and these interactions can be covalent peptide bond, they can be hydrogen bonds, they can also be disulfide linkages between sulfur containing amino acids, and also they can be van der Waals interactions. So all these interactions actually help in stabilizing the protein and formation of its structure. And guys, as I said, the structure of protein is very important for the function. So basically the 1-degree structure, 2-degree structure, 3-degree or the 4-degree structure of protein actually decides the role of the protein. Let's say hemoglobin. So friends, hemoglobin transports gases, right? Oxygen in and carbon dioxide out. And this function of hemoglobin is performed because of this unique structure of hemoglobin. Friends, let's first focus ourselves on the simplest protein structure, which is the primary structure. So guys, the primary structure is the basic or the simplest protein structure which is formed just by bonding between various amino acids. So, a straight chain structure formed by bonding between amino acids is the primary structure. The primary structure tells us about how many polypeptide chains are involved in the structure. It also tells us about the number and the sequence of amino acids in the structure. Now guys, if we look at the primary structure, then friends, the left side of the primary structure is actually representing the first amino acid of the chain. So, this left side amino acid is the first amino acid, is also known as N-terminus or amino-terminus amino acid. Right? So, this is the N-terminus of this primary structure. And the right side actually represents the last amino acid and is also known as C-terminus or carboxyl terminus amino acid. So friends, if we have a look at this particular structure, then here clearly you can see that in A chain, the glycine is the first amino acid, so this is the N-terminal amino acid. And asparagine is the last amino acid and it is the C-terminus amino acid. While the sequence is different in the B chain of this protein primary structure. We can say phenylalanine is the first amino acid or the N-terminus and alanine is the last amino acid or the C-terminus. So with this we are pretty clear with what is the primary protein structure. Friends, the primary protein structure actually tells us about the sequence of amino acids and also number of amino acids in a chain. So this basically explains us which amino acid is present where in the primary structure. Friends, this sequence of amino acids is actually coded by the sequence of nucleotides in the DNA or the genetic material. So it is the DNA which basically decides the sequence of amino acids in a protein. Friends, any single change in the nucleotide of DNA will cause a change in the amino acids. So if a single amino acid is changed, then overall protein gets changed. Thus, protein structure changes, and if the structure changes, the function automatically changes. And this thing, friends, can sometimes have a serious consequence on the body cell. So, everything happens in cases of protein very precisely. The primary protein structure usually has covalent bonds like the peptide bonds in them. So, peptide bonds are in majority, while sometimes disulfide linkages are also present in those protein structures in which cysteine is present as amino acid. Now guys, the primary protein structure is generally the non-functional form of protein. Yes, generally primary structure is non-functional. However, the exception is the insulin molecule. The insulin molecule is functional in its primary structure and it is made of some 51 amino acids. And guys, the structure we took as an example for primary protein structure was actually this insulin molecule. This primary structure is the first stage of protein formation. So higher levels of protein structure are largely dependent on the primary structure of those proteins. So guys, any protein, may it be an enzyme, a hormone, also muscular proteins or any type of protein for that matter, the function of the protein is largely dependent on the primary structure of that protein. Isn't protein structure a fascinating area to study, guys? Of course it is. But here you must be thinking that how do we move forward from the amino acid sequence which is present in the protein primary structure to the three-dimensional structure of a mature functional protein. How will we get this one? Well, this is what we have to explore in our future sessions, and let's now quickly summarize whatever we have learned about the primary protein structure in this session. Peptide bond is a covalent bond formed between two amino acids by condensation reaction and removal of water molecule. Proteins are made of one or more polypeptides and they show 4 levels of structure organization: primary structure, secondary structure, tertiary structure and quaternary structure. Primary protein structure is the simplest structure. Primary structure has long linear chains of amino acids joined by peptide bonds. Primary structure has a specific sequence of amino acids. The left side in a primary structure is the first amino acid or the N-terminal amino acid, and the right side is the last amino acid or the C-terminal amino acid.